So the superficial or the top layer of the skin is the epidermis, and below that there is a thicker layer called the dermis. So the dermis is the thicker layer of skin below the epidermis. And this is a very tough tissue, and it's described as a moderately dense connective tissue. It's moderately dense. This means it's got quite a few fibres running through it that make it a little bit dense. And we've said it's very tough. And the reason it's tough is that it contains collagen, strands of interlinked collagen. Collagen is a protein with very high tensile strength. It occurs in strands and it gives the skin its strength. It's tough stuff. And if you don't believe me, just look at some leather, because leather is treated dermis. The collagen makes it very strong. But as well as being tough, it's also very stretchy and very elastic. And this is because as well as containing collagen fibres, this moderately dense connective tissue also contains elastic fibres that give it its elasticity. So it's stretchy and it springs back after it's been deformed, and it can bend around the joints of the body. So it's tough, but it's also elastic. It's a moderately dense connective tissue. Now, these collagen fibres and elastic fibres are all produced by fibroblasts. Fibroblasts are cells that produce these fibrous tissues. A blast cell is a cell which produces something. So the collagen and the elastic fibres are produced by the fibroblasts. And all the little spaces in between the fibroblasts and the collagen and the elastic fibres are all filled up with another substance called ground substance. Now, ground substance is also sometimes called matrix. Matrix and ground substance are exactly the same thing. And it's basically a glycoprotein. It contains glyco is carbohydrates, glucose type molecules, and protein type molecules. So the fibroblasts are producing this glycoprotein ground substance that fills in all the gaps. And the ground substance and the collagen also attract some water, which gives the dermis a three-dimensional structure. It's called turgor. It's what gives it a body. Because when patients are dehydrated, you can see that this body is lost and the skin loses this turgor. And when you bunch bits of it together, it can stick together because it's lost the water in it that gives it its normal body. So we're dealing with a moderately dense connective tissue with collagen and elastic fibres and fibroblasts and glycoprotein ground substance and matrix. High tensile strength, but combined with elasticity. Now, as well as the fibroblasts, there are some other types of cells in the dermis. There's macrophages, for example, and dendritic cells. Macrophages and dendritic cells are both immune cells. They both derive from monocytes with an immunological function. And there's also mast cells as well. Mast cells contain granules that contain such chemicals as histamine, and they will facilitate inflammatory reactions should an inflammatory reaction be required. So if a tissue is injured, you want an inflammatory reaction to facilitate healing of that tissue. So the mast cells can facilitate inflammation so there can be healing if the dermis is damaged. And as we noted in the podcast on the epidermis, the junction between the dermis and the epidermis is wavy. There are projections of the dermis rise up into the epidermis, and these are called dermal papillae. Papillae means nipples. They're like nipple-shaped structures that rise up into the epidermis. And this gives a large surface area between the dermis and the epidermis, because the oxygen supply and the nutrient supply for the epidermis come from the tissue fluids of the dermis. So there's this papillary region of the dermis, the top part, and the strands of collagen in this top part are relatively thin. But then deeper down in the dermis, there are thicker layers of collagen, that form a reticulum. A reticulum is a network. So this area of the deeper dermis, where there is a network of thicker strands of collagen, is called the reticular region of the dermis. So there's the papillae region on the top, which is a relatively thin part of the dermis, and there's the deeper reticular area of the dermis as well. So that's the nature of the tissue of the dermis. But of course, as well as the tissue, there are specific structures that go in the dermis. So there's blood and lymphatic vessels, for example. There's nerve endings. There's hair follicles and sebaceous glands, sweat glands. There are these specific structures within the tissue of the dermis. So let's go on and think about those in the next part of this talk. Now I think we'll start off by talking about the blood vessels. Some of the blood vessels in the deeper muscle go through the fascia of the muscle and into the skin to supply the skin with its blood supply. And other blood vessels run along in the hypodermis as well and branch up into the dermis. But however the blood actually gets into the dermis, it divides into small blood vessels called arterioles. And these arterioles will divide into beds or groups of capillaries. The blood will go through the capillaries, and the capillaries are very thin-walled vessels. The thin walls of the capillary allow gaseous exchange, allowing oxygen to go from the blood in the capillary into the tissues, and carbon dioxide to go from the tissues into the blood. It allows nutrients to go from the blood into the tissues, and waste products to go from the tissues back into the blood. And then when this has taken place, the blood goes from the capillaries through into a venule, into a small vein called a venule. And several venules will join together to form larger veins, draining the blood back into the systemic venous circulation. So blood's going in an arteriole, through a capillary network, and it's going out via a venule. And there are lots and lots of capillaries in the dermis. Now the arteriole, before the blood goes into the capillaries, around about the wall of the arteriole there's a sphincter, and it's called the precapillary sphincter. And this can contract or it can relax. When it contracts, that's called vasoconstriction. When it relaxes, that's called vasodilation. So for example, when it's cold and you don't want lots of blood to go near the surface of the body, it's going to constrict. There's going to be peripheral vasoconstriction. That means less blood will get into the capillaries, therefore the skin will look pale. It will develop pallor. It will be a paler colour because there's less red blood going through the capillaries. But then, for example, if you're too warm, the capillary sphincter, the precapillary sphincter, will dilate. You'll get peripheral vasodilation, meaning that more blood goes into the capillaries. And that means the skin will have a redder type appearance. And sympathetic stimulation, that is stimulation of the autonomic sympathetic nervous system, will cause vasoconstriction, giving rise to pallor. And when there's a lack of sympathetic stimulation, the blood vessels will dilate. And also if you drink alcohol, that will cause peripheral vasodilation and make you take on a reddish type of appearance. So lots of blood going through the dermis, through this rich network of arterioles, capillaries and venules. Okay, so next I think we'll talk about the role of the dermis in sensation, the sense of tactility. Now we've already noted in the podcast on the epidermis that there are Merkel cells in the very deepest layer of the epidermis. They're the only nerve fibres that go into the epidermis, and they just detect light touch, very light touch. So there's the Merkel receptors actually in the epidermis. But all of the rest of the sense of tactility, the nerve fibres for that are in the dermis. And these are peripheral sensory detectors. Now what these do, when there's an external stimulus on the skin, these detectors detect that. They are mechanoreceptors. They detect this mechanical stimulation. Or they're thermoreceptors. They detect the temperature. But whatever it is from the outside world affecting the skin, they detect it. And when they detect the particular sensation, they generate a nerve impulse that represents that sensation. And that nerve impulse then goes off towards the brain. So, for example, if it's in part of the body, that peripheral sensory receptor will be connected to a dendrite of a sensory neuron. That will go to the cell body of the sensory neuron. That will go into the spinal cord. That will go up the spinal cord to the brain. At some point it will cross over to the opposite side of the brain, and that sensation will be then interpreted as a somatic sensation in the sensory cortex of the parietal lobe in the postcentral gyrus. The postcentral gyrus is the part of the parietal lobe that contains the sensory cortex, where all tactility is experienced. So what particular sorts of sensors are there? Well, really there's two types of peripheral sensory receptors in the dermis. There's those which are just free nerve endings distributed throughout the dermis, and there are those which are associated with particular sensory corpuscles. So let's think about the ones associated with sensory corpuscles first. Now, the first type are typically found very high up in the dermis, in the superficial dermis, just underneath the epidermis. And these are called Meissner corpuscles. Meissner corpuscles. I think they're named after some German anatomist called Meissner. Anyway, what they detect is touch. So they're near the surface, so they detect touch. That makes sense. They're near the surface. But deeper down, there are other sensory corpuscles called Pacinian corpuscles. And the Pacinian corpuscles are deep down in the dermis because they detect pressure. They are pressure detectors. And wrapping around the hair follicles, there are groups of nerves called the hair root plexus, and they detect very fine movements in the hair follicle. And of course the hair shaft runs through the hair follicle. So if the hairs are just brushed on the surface of the body, even without touching the epidermis, that's going to flick the hairs. The hair shaft within the hair follicle is going to be flicked and moved, and that is detected. So this is a very, very sensitive type of detection, because you can just have the slightest brush on the hairs without touching the skin itself, and yet that sensation can be detected because of this sensitive hair root plexus. And then, as well as these peripheral sensory receptors, there are other peripheral sensory receptors which just appear to be free nerve endings branching through the dermis. They're not associated with any particular corpuscles, as were the Meissner or the Pacinian corpuscles. And this is true for the nerve fibres that detect heat. So some free nerve fibres detect heat. Other free nerve fibres detect cold. So there are hot thermoreceptors and there are cold thermoreceptors. And they're just free nerve endings in the dermis. And another type of free nerve ending detects pain. The nerve endings that detect pain are called the nociceptors, and they are activated at the point where the tissues become damaged, so we get a pain alerting us to the fact that the tissue is becoming damaged. So there's touch, pressure, heat, cold, pain. The other sensations that the dermis can generate are itch and tickle. So, I don't know, do you still get ticklish when you're an adult? Maybe to some extent. Children certainly get it. And tickle is detected by free nerve endings in the dermis. And itch is also detected by free nerve endings in the dermis. And when you're in clinical practice, you can come across patients with the most appalling itchy conditions. For example, patients with allergic reactions can get intolerable itches. They're almost like ripping the skin off. because of the inflammatory mediators stimulating the itch receptors. And the other very, very itchy group of people that I've come across are patients with jaundice, because the bile salts are in the dermis, making the patient look yellow, and the bile salts are also stimulating the itch receptors, causing really, really severe itching. So they're the different senses of tactility generated by peripheral sensory receptors in the dermis, but experienced in the sensory cortex of the brain. Heat, pain, pressure, touch, itch and tickle. So let's just think where we're up to. We've done the dermis as a connective tissue, we've looked at the blood vessels, we've looked at tactility. Let's now consider the sweat glands. Now, the posh name for the sweat glands is the sudoriferous glands that produce sweat. And there's actually two types of sweat gland in the skin. There's eccrine sweat glands and apocrine sweat glands. Let's think about the eccrine sweat glands first. Now, the eccrine sweat glands are those that facilitate thermoregulation. So when we start getting hot, first we peripherally vasodilate. If that doesn't cool us down enough, we start sweating. Because when sweat is deposited on the surface of the body, it evaporates. And the heat it needs to evaporate, it extracts from the surface of the body, therefore removing heat from the surface of the body. This is called the latent heat of vaporization. And to evaporate just a little bit of sweat uses a lot of heat from the surface of the body, therefore cools the body effectively. So if people can sweat and are still producing sweat, they can normally manage to maintain body temperature. But if people can't sweat, or that sweat cannot evaporate, then they can become hyperthermic, and of course that's a life-threatening condition. Or if someone's dehydrated and can't make enough sweat, again they're not losing the latent heat of vaporization from the surface of the body, so they can get life-threatening hyperthermia as well. Maybe the most common cause of this actually is people wearing too many clothes when it's hot, so the sweat can't evaporate, because it's only when the sweat evaporates that the heat is lost from the surface of the body. It's sometimes very important to understand that. Anyway, this sweat is produced by the eccrine sweat glands. Now, these have a glandular unit, which is a coiled tube, usually very deep down in the dermis, and they also have a sweat gland duct rising up through the dermis all the way through the epidermis, depositing the sweat on the surface of the body. So there's a glandular portion producing the sweat, and there's a ductile portion taking the sweat to the surface of the body. And I don't know who's counted them, but some books say there's over 3 or even 4 million eccrine sweat glands on the surface of your body. Interesting idea. Now, sweat contains water mostly, and sodium chloride, sodium and chloride, salt in other words. And I'm sure you've noticed that sweat tastes salty. And also there's some waste products in sweat, like there's some uric acid, some ammonia and some lactic acid, and urea can be excreted in the sweat. Now, even although these things are in sweat, they're only in sweat because they're in the plasma. This is not a homeostatic sort of excretory organ. The sweat glands aren't really excretory organs like the kidneys are. It's just that the constituents of the sweat will reflect the constituents of the plasma. So some of these things will be lost. But there again, some useful things are lost in sweat as well. For example, there's some amino acids and some glucose in sweat. So it's reflecting the constituency of the plasma really, rather than being an excretory organ. The main excretory organ, of course, is organs are the kidneys that get rid of these things in a homeostatic way for us. So sweat is produced when we're too hot, And sweat is produced as a result of sympathetic stimulation. So when you're anxious or excited or during exercise or in emergency situations, when the sympathetic nervous system is stimulated, then you can sweat as well. So when you're anxious, for example, we call this a cold sweat, don't we? You're sweating even though you might be cold because it's a sympathetic activity. So that's the eccrine sweat glands. But there are these apocrine sweat glands as well. And these are larger sweat glands, and they only function from puberty. They don't work in children. And the secretions they produce are acted on by bacteria on the surface of the skin, which gives rise to characteristic smells. And these apocrine sweat glands are mostly found in the axilla, that's the armpit, round about the groin. But they're also found around the areola of the nipple, that's the coloured part around the nipple in women. And they're also found on the bearded area of the face in men. So it's speculated that they act as pheromones. Pheromones are sort of hormones that work between organisms, between individuals. So part of the reason you might be attracted to someone is because of the way they smell. You might not realise it, but their smell can be affecting your brain, giving rise to feelings of attraction. And the other thing about the apocrine sweat glands is that the ducts open into the hair follicles. So the sweat actually comes out of the hair follicles. Anyway, they're not thermoregulatory. They seem to have this much more hormonal type of function. And just to finish off sweat glands, it's probably worth noting that the ceruminous glands open up into the external auditory meatus. That's your ear hole, and they produce earwax, which protects the outer part of the ear. So we've mentioned the dermis, blood supply, tactility, sweat glands. Let's think about hair follicles next. Now, hair follicles are deep structures that go down into the deep dermis and even into the hypodermis. And they are downward projections of the epidermis. So in fact, keratinocytes, that is the epidermal cells, line the hair follicles and form the base of the hair follicle as well. And at the base of the hair follicle, there is a hair bulb. Now, there's a capillary inside this to give it a good blood supply. And in the hair bulb, the keratinocytes are rapidly dividing, being pushed up. They keratinize and flatten. And in combination with some extracellular protein, you end up with stacks of dead keratinized epidermal cells with this protein sticking them together. And these form the shaft of the hairs. And as the hair grows, this is pushed up. And that's how the hairs grow. So the hairs are growing out of the hair follicles. And connected to the hair follicles, there are also hair erector muscles. And when you're cold, these hair erector muscles will contract. And this is what causes goose pimples when we're cold. The idea is that the hair stand up on end, and that increases the thickness of the boundary layer, trapping some air next to the surface of the body, because air is a very good insulator and will help to keep us warm. Sometimes the hair erector muscles are called the erector pili, but it means the same thing, the hair erector muscles. And also opening onto the hair follicles, or opening into the hair follicles, there are the ducts of the sebaceous glands. Now, sebaceous glands produce a thick, oily substance called sebum. So sebaceous glands produce sebum. And this sebum goes into the hair follicle, rises up the hair follicle between the hair and the follicle, and it goes onto the surface of the skin, as well as lining the hair, and it's waxy and oily. And it makes the surface of the skin waterproof and supple and stops it from cracking out. It's an oily, lubricating substance. And again, the amount of this is increased under hormonal influence at puberty, which is why people can sometimes become a little bit greasy during puberty. And if you want to feel some sebum in you immediately, just feel in between your nose and your face, and you can feel some oily sebum in there. But there is some sebum all over the surface of the body. It helps to waterproof and protect the surface of the skin, which of course is normal and good. Now I think I'll maybe mention one more thing. In the dermis there are also lymphatic capillaries, and the lymphatic capillaries drain into small lymphatic vessels. These are called afferent lymphatic vessels. They drain into progressively larger lymphatic vessels, which in turn drain into lymph nodes. And these will drain away bacteria which get into the tissues. And also, if any bacteria or viruses get into the dermis, that will be detected by the dendritic cells in the dermis. These are sometimes called antigen presenting cells. So if an antigen gets in, that will become associated with the membrane of the dendritic cell. The dendritic cell will then be stimulated to move into the lymphatic vessels. That will move off to the lymph nodes, and in the lymph nodes the dendritic cell will act as an antigen presenting cell, and it will present that antigen to the B and T lymphocytes, which will then mount a specific immune response. So it's very important immunologically that the dermis has lymphatic capillaries and afferent lymphatic drainage vessels. And as well as that, sometimes you get a little bit of protein leaking out of the capillaries, and you don't want the proteins hanging around in the tissue spaces because the proteins are very osmotic and it will cause edema. So again, when proteins do escape from the capillaries, they're drained away in the lymphatics, which of course are eventually drained back into the subclavian veins once they've passed through the whole lymphatic system in the body. So I think that'll do for this review of the dermis. We've talked about the dermis as a moderately dense connective tissue, the blood vessels, the peripheral sensory receptors which give rise to tactility, the pseudoiferous or the sweat glands, the hair follicles and the hair. We've talked about sebum, and we've talked about the lymphatic vessels. Actually, I think I might just mention that underneath the dermis there is the hypodermis, the layer under the dermis. And this is mostly, in most parts of the body, it's mostly adipose tissue. It contains adipocytes, which are the fat-containing cells. And the adipose tissue in the hypodermis is actually described as a loose connective tissue. So it does contain some fibroblasts, it does contain some strands of collagen and some elastic fibres, but not as many as in the more dense dermis. So the dermis is described as a moderately dense connective tissue, whereas the hypodermis is a loose or a less dense connective tissue.